So welcome to Yale. It's good to see people in person. So it's my pleasure to introduce Professor Lucila Ohno-Machado. She's the Waldman-Vossaitis Professor of Medicine, Biomedical Informatics, and Data Science. She's also a Deputy Dean for Biomedical Informatics, and she chairs the new section of Biomedical Informatics and Data Science. She received her MD from Sao Paulo in Brazil. Then she went to Stanford to do PhD in computer science and informatics. And then after that, she spent time as faculty at Brigham and Women's in Harvard for a few years. Then she crossed back over to the West Coast to go to San Diego to start a new department of Biomedical Informatics. And then it's been a luck to get her to come back to the East Coast to start the same thing at Yale. So she has many accolades and awards. I'm not going to go through all of that. She's a member of the National Academy of Medicine. She's the principal investigator of the All of Us grant that's trying to record data from over 1 million people, both social and medical data. So, I mean, usually when you do introductions, you try to figure out what to say about a person. But if I want to take my time, you wouldn't have time for her talk. So I'm going to stop here and let her start with the presentation. I will switch the slides here.
Thank you so much. And thank you for coming, and those online as well. Thanks to organizers. I've been here for almost 3 months now, so you can ask me anything about Yale, and I will refer you to somebody else. It's a great pleasure to be here. I think I will every 5 minutes say this is the deep reconstruction workshop, because otherwise you will think you're in the wrong place, since I will talk nothing about deep reconstruction in my talk. It's mostly a presentation of what our unit came here to do, where we will have imaging experts. But for now, I'll talk primarily of work we have been doing in the clinical informatics space, and also something that is very important for all of you as you collect these images to analyze is the diversity of the population from whom you are gathering this information. So I'll talk a little bit about everything here, but primarily introducing our discipline and introducing our new section, soon to become a department. So let me advance here. Again, the objectives, I hate to say, but I don't have any conflicts of interest, unfortunately. No financial ties to anything. So I'll talk about biomedical informatics and biomedical data science, describe a few examples, and how statistical and machine learning have been used to predict certain clinical outcomes, and introduce the problem of evaluating personalized predictions, which at one point will be your problem too. So hopefully that part will be more applicable to what you're doing, and then provide an overview of this important cohort collection that we'll start here at New Haven as well. So in terms of biomedical informatics and data science, what I see is the end goal and the vision of making human health and patient interactions better. So mitigating the effects of disease, perhaps preventing disease, and in general, having people just be healthier. There are a lot of boxes here. I won't go over them, but it's about the subspecialties of informatics that are involved in order for us to get to that point. And it's a circle. So it goes all the way to creating decision support systems that can be applied in the clinic. And then you collect data all over again and try to do the circle. So many things are involved. I'm particularly involved in data de-identification and in evaluation methods for predictive models, but my colleagues are in the other areas here. So as you can see, it's a lot of different areas to cover. The types of health sciences, big data, as you know, there are a lot of them. You happen to be in a very difficult one, which is imaging, but also in signal processing, genomes and transcriptomes, there's a lot of data analysis going on. So I'll talk a little bit about what's the vision for the healthcare, what's the best for each patient, as opposed to what is best for each disease. And so that's the notion of personalized care. What's the influence of environment, privacy, and molecular level findings, cost-effective care. So there are many topics that again cover that circle that I presented in the beginning. And where we stand as biomedical informatics and data science is at the intersection of what you could call bioinformatics or computational biology, the medical or clinical informatics, and then public health informatics as well. A common set of algorithms, tools, controlled vocabularies, ontologies, data management practices, and information retrieval techniques are involved. So I'll go through the history of it, but don't worry, because it will be very quick. This is in the 1960s where the first units of computer science appeared in hospitals, and in this case, in Mass General Hospital. And if you look carefully, there are wires going across the terminal, and that's the touch screen of the time, is how you could select options at that. And I said I'll go quick because 20 years later, this is the first graphical interface of a Macintosh computer at Brigham and Women's Hospital, also in Boston, and where they were doing training of medical students. And the big thing was not only a black and white graphical interface, but sound, because computers didn't have sound up to that point. So you go 20 years even later, at a combination of Harvard and MIT, we developed this scalable medical alert response technology, which was in the days that if anyone thinks that is a cell phone, it's not. It's called a personal digital assistant, a PDA. And it was like, we were very excited about it because it has some computation power. So my colleagues at MIT, they adapted for it to be an EKG, a local EKG. And we also did indoor location with an added ultrasound-based device. So we could monitor patients in the waiting area of the emergency department, because many times some event happens there and no one is monitoring. So this is with this simple EKG and the location device, we could do an alert system. Again, everything from scratch, which was quite interesting time. Now you go 20 years later, oops. And that's what we were doing at University of California, San Diego. A colleague of mine, very interested in sepsis prediction and the interfacing ICU devices so that he could, within 4 hours earlier than typical, predict the development of sepsis. And the reason this is important is because if you can intervene earlier, the sepsis either will not install or will be much milder than otherwise it would be. And in addition to that, we started other investigations, or like, for example, how to use blockchain as a immutable ledger and smart contract technology. And clinical data networks. So a lot of interesting things. Our field is good because you get interested in a technology and you can always insert some healthcare or biomedical domain into it and study at that point. So I'll go a little bit out of order because we have, to care about data. We have to care about models and implementation. I'll start with the models because that's where I can talk about the privacy, but then I'll briefly touch on the two other items as well. So statistical and machine learning models, they're not new, and they have been used for healthcare before. It's just now they have much more attention. And I would say some models are statistical learning, or they're regression models used for various things. One of the most known uses is for the transplant queue for liver transplantation. It's not a first come, first serve. It is whoever has the highest severity. And the severity index is calculated by what used to be the Mayo model for end-stage liver disease. So from the Mayo Clinic, they called just model for end-stage liver disease. And that is how the transplant queue currently runs. Other examples in acute kidney injury, severity indexes at the hospital and so on. And many are available even in the electronic health record, EHR vendor products. So the one used here is called Epic. But the problem being local evaluation is rare, and that has some problems associated with it. Because the adaptation of models to the local population is important. These models were developed in a population that is very different from the one you were applying to. You will get, you can do harm. You will not be doing the right thing. And so there's a huge opportunity here at Yale to connect every single department with informatics to develop predictive models in several specialties. But as you know, AI requires access to large data repositories, and also aggregating data from different countries in different populations becomes quite important. The number of observations is way higher than those that we used to go with for regression. So with that comes the problem of sharing data and protecting the privacy of patients, right? With small sets, it happens too, but now we're talking large scale privacy breaches that could happen. In clinical data sharing, so sharing of clinical data, there is a dilemma. Is it right to share the data? Because the patients are not always aware that this patient, their data, your data, is being used out there to create predictive models. Is it right not to share? Because there is knowledge that can be acquired from the sets, and the sets are so-called de-identified, so the risk is reduced when identifiers are not shared. And then we have done work. Can we share without moving data around? Can we at least keep data in their own places so you don't have to give away a large set of data that someone can mine and try to re-identify? So these are all things that are happening right now with large cohorts of patients. And I'll just give you an example here, but because it's very simple, everyone knows that biometric, so everyone knows names would not be given, right? In de-identified data sets, you remove the names. But if you include a biometric there, what's the problem? The problem is then I have a target individual who I get the biometric from, and I check against the rows, and then I find, oh, Lisa is pregnant. The person I know matches that and read the rest. So biometrics are considered identifiers. Of course, they're the best ones, and they're removed. But that are other problems too in the clinical areas. If you're doing clinical records and using them in potentially sharing with other people. So this table in the middle without the names again would be considered de-identified, right? But if you know the combination of A equals 10, B equals 20, and you know Lisa signed up for that study, the easiest thing is to do the same thing, just match Lisa against the data set and read the rest of it. So it's always a trade-off, right? The information you have and how you can link to the table and how you can read the rest of it there. So there is risk of finding something about the person of interest. And in this case, it's unique. So you knew to go to that role, right? Because you matched Lisa, but it doesn't need to be unique. If all you wanted to know was the diagnosis, it doesn't matter if she earns 60K or 20K, you're not interested in that part. And you would still learn something about the diagnosis of this person. So you're probably thinking now, well, then it's impossible to share data because there will always be this risk. And the answer is yes, it is. There will always be risk. There's no way to say if data have utility, if they're good for some predictions, they will have the risk associated with them as well. So for my colleagues in genomics, it's the genomics are biometrics. They are very good biometrics, right? So anytime you have a study that gets you access to the genome of a person, it doesn't need to be the whole thing, just certain parts is sufficient. And some other information. that other information is at risk, right? Because anyone could get my water bottle, sequence my genome, and then go against every single database in the world and see if I contributed to any research and read out the remainder of the record. So again, HIPAA is the Health Insurance Portability and Accountability Act. And it is to regulate how electronic health record data can be used. And if you go to the doctor, you do sign a HIPAA agreement that your data will be used for that. So just wanted you to know that genomes are like biometrics. And what about, and again, uniqueness does not guarantee privacy. So genomes are identifiers or can be used as identifiers. What about images? Images can too, right? And it, you know, is the same issue here. And I don't know, several years ago, 3 years ago, probably, there was even a paper that would reconstruct faces given the genomes. And that was also, you probably know about that. I don't, you know, I don't have the skill to know how good that reconstruction was, but it's scary. If you can get a genome and reconstruct the face of the person, that's pretty interesting. So again, genomes are much better identifier than fingerprints, right? Because they bridge your privacy and they bridge the privacy of your family as well. So the way the Golden State Killer, not that we are proud of it in California, but that's the Golden State Killer, who was only caught many, many years later because a cousin had contributed to a particular ancestry database. And then it was found. So very good use, you know, very good thing that happened, but it opened a debate about DNA privacy and also of people wanting or not to contribute to this databases, because in a way you're making a decision for yourself and for your blood relatives too. So privacy protection solutions we want to share, we want research to have access to a whole lot of data, but we are a little, you know, we're careful about that. There is federated computation that at least you keep your data local, you don't give all the data away, and some distributed analytics. So the goal here is the user, the researcher asks a question, the algorithm decomposes the question, gets answers in different places and comes back to the researcher, but they never shared the data, their data stayed separate. So I believe the next slide is the only one you need to remember here on why this is a good thing to be done. And this is it. You just memorize, so ask about it later. No, but the whole thing here is just to show that the sums are per site, right? So you have a site that has some data, has another site that has some data. If you can, like you can for a regression model, know that this sums can occur in each data set, then why would you ever need to put all the data together? And the answer is you don't need, right? It's just like parallel computing, the answer. So some algorithms you can do that without approximations. Some others you need approximations, but the idea here is you distribute the algorithm and you don't need to move patient data around, which confers some privacy, but of course you can breach the privacy wherever you in the other places. What I've been describing primarily is same type of data of patients who are in different systems, different hospitals. So you can, you know, virtually aggregate them and have a larger data set. But there's also an interesting problem when you have the data from the same patient distributed in different databases, and you still want to do this multivariate calculations without putting the data together. So it's a more difficult problem. And again, there were several folks working on that, but it's very real. If you think the genome centers will retain the genomes and the hospitals will retain the clinical data, then it's exactly this type of computation that you'll be looking for in order not to send data around. And we are currently working on this two large cohorts, the Million Veteran Program and the All of Us Research Program, which is funded by the NIH, which right now has almost 500K participants, 200K genomes already available. Unfortunately, no imaging data so far, but if you're interested, it is an important thing. The UK Biobank has imaging data, as you know, but all of us not yet. So we need to do something about this. And what we're working again is on secure federated algorithms, so you can do this computation without centralizing the data. But now let me talk about something that is very important, which is the data themselves. So if you were following AI fairness movements and so on, and so there are techniques to try to fix the distribution of the data, because you only have a majority of this particular demographics and so on. So that's definitely something to be done, but why not fix the people, get the right people to get the data from? That's way better, right? Because all these other fixes are either you replicate the folks who were less represented just to look, but that is the same person, right? You're not varying too much. So the better things is to get the diverse people in the beginning. So we do studies on clinical data networks, in which, again, we get a data from different modalities, and we try to, again, diversify as much as we can in each one of these aspects here. So the All of Us research program was funded 5 years ago by the NIH, and is an effort to increase diversity in the collection and to be a large cohort from which hopefully other cohorts, sub-cohorts can be derived, in which you have the genome sequencing, you have survey data, you have a whole lot of other data from the participants, and then possibly can do parallel research and do more in depth in certain areas. It has a biobank and that you can order specimens from, or will be able to. But it, like I said, unfortunately, it does not have images yet. It's a national open resource, so the data are to be available for use. So again, the data don't leave the computational enclave of all of us, but are there to be used. So these are the types of data they are there right now. I won't go into details. Wearables on digital apps are a smaller collection, but the others are quite comprehensive at this point, and the program is midway into its 1 million goal. We in California developed that 1/10 of the recruitment is coming from our consortium in California that I'm still co-leading with a colleague, and there are several others, as you can tell, contributing to this collection. The interesting thing that will happen here, starting in June, is the collection at the YCCI, the Center for Clinical Research, which had partnerships with the community and also with Puerto Rico, and we will have a site collection there. So it's exciting because, again, it's patients from our system that will be going to this process. You can say, well, but it's still very skewed towards white patients in a way, but if you look at it, other studies do not have this many representatives of Black African Americans or Hispanics and so on. So even though it's targeted towards this groups, it's still, you know, a growing number. And I would draw your attention to more than one ancestry there. It says just 6.5%, but that's a matter of how you measure right? Because of course, almost everyone is mixed, but the degree of mixing will determine whether people auto-report as mixed or not. Now, the research hub components. So these are all data to be used for research by anyone. There are simple ones, there are command line ones that you use R or Python to, again, pose the questions you want and work on this researcher workbench. So just as a plug, on March 29, Dr. Jeff Cohen will talk about how do you use this data set. So it's a local Yale investigator who has done several papers using this particular data set. And this, again, we set up our network there because we had pre-existing clinical data networks in which we had the University of California for the first time virtually aggregate records, as well as our rivals in other cities or other institutions in the VA, the whole VA system contributing to analysis in this way. You know, we never get their data, they never get our data, but we can consult across. In the beginning of the pandemic, when we were all home thinking of what to do, we thought, let's put this network to work and try to answer questions that were coming and no one knew at the time. You know, the handling of patients with COVID-19 was still a new thing, and a lot of questions came up. One of the questions, and this is our clinicians put questions, if we can answer with clinical records, we would. Again, using this network that has all the VAs, plus a few of our friends in particular health systems that were contributing to this effort, including one in Germany. So in the beginning, again, there were very few patients we could do analysis on. There were only 2,500 deaths in the system at the time. So you see how difficult it was to get definitive answers to anything. One of the first questions was about ACE inhibitors. And this is a type of antihypertensive medication. People thought, because of the way the receptors for the coronavirus work, that people taking this particular drug would have higher complications. And we showed that no, apparently, at least in terms of mortality, it wasn't higher. It was actually lower than some other antihypertensive medications, and definitely better than not than stopping the medications if you are hypertensive. So think about it, we're even though hypertension is very common, we had to get the hypertensive patients by a certain definitions who were hospitalized and then calculate the different rates in there. So if you had just one hospital, it wouldn't work. Even if we had the whole VA, it wasn't numbers enough in order to get to that conclusion. So we had at that time 10 institutions contributing some data, and subsequently this was confirmed with studies. The other thing is to do this multivariate model across the network too. And the other thing that we showed, even though it was said, and it was the statistics at the time, that there was a higher mortality among Black patients and Latinos. If you just looked at the hospitalized one, they were having the same outcomes. It wasn't a factor into mortality. The age was the big factor at the time. But of course, you can tell why the statistics would be the other way, right? Because you may have fewer access, for example, or lower standard of care for certain in general. But once you are in, you know, in the health system, and this health systems are operating well, there was no difference essentially. So I talked about these two parts were super important, and then there are implementation issues too. So I would just go over very quickly because evaluating predictive models is also something that my machine learning friends don't pay too much attention to. And that's alarming because if you're using that and giving that information either to a clinician or to a patient, you've got to know what you're doing, right? You can't just say, oh, your probability of dying is 61%, 61.3399. That's what you get from your algorithm, right? You can get that, it seems very precise, but it's a thing that is very hard to evaluate. So areas under the ROC curve, super popular in radiology, right? People have developed a lot of methods and so on, but they only go so far, and I'll explain why. So the whole concept of areas under the ROC curve in this toy example here, you have on the left-hand side for patients who were gold standard alive, what the predictions of the algorithms were sorted. And on the other side, same thing for the patients who actually died, and you made predictions as well. And if I give you this data, just that table, you will calculate an area on the ROC of 78.5%. And I always had some trouble understanding what that meant, but there is an interpretation that is very simple. The area under the ROC curve can be calculated as the number of concordant pairs across these two columns, plus half of the ties, divided by all pairs. So I have 10 patients who died, 10 patients who were alive. There are 100 pairs there, right? And I'm just showing the discordant pairs. I'm telling you it's 21 of them, and I have one tie, right? They are discordant because I gave a higher probability of dying for someone who didn't die than for someone in the pair. So that's how you calculate. It's the same way. C concordance index is the area in the ROC curve. And why do I care? I care because areas under the ROC curve, when people publish, say, oh, when I validated in another system and the area in the ROC curve is the same, right? There's no statistical difference. Yes, that's a good sign, but that's not the only thing, right? That still doesn't mean it's a good model to use in that particular case. And I'll tell you why. Because you divide all estimates by 10, right? And that's the probability of dying. You get the same area under the ROC curve. So it doesn't give you the calibration for the estimate as you would need if you're talking to a person. Because of course I would prefer to have a 0.007 probability than 10 times more. But again, is the model calibrated or not? It's what you have to think about. And there are ways to recalibrate models so that you get closer to what we think, would be the case. So I won't go through those details, but I just wanted to give you that, because reviewers up to these days still don't get this notion that you have to have more than an area under the ROC curve improvement in order for a model to be useful. So you can use errors. There are several types that you can use and so on to just, again, demonstrate the difference between these three scenarios, which actually would give to each person a very different probability. So that's one thing that happened, for example, in a model of acute kidney injury that we did using data from UCSD. But then we tried to validate on Mayo Clinic. And here's the calibration plot where you're supposed to have every group kind of in the diagonal. But you see that in the Mayo cohort, the model very much underestimated the probabilities. So meaning it's a different population in Mayo or in San Diego. And unless you recalibrate it, you shouldn't be using that model, even though, same area, the ROC curve and so on. So I won't get into more details, but it is an area that I always found very interesting, because it's very hard, to what is the real probability, right? It's very hard to say, oh, the real probability is this or that. I must just conclude saying that risk prediction models are being done, tons of them. These are just look how many models were included in certain systematic reviews, and this is for acute kidney injury only. They're for chronic kidney injury, they're for many other things. Of the more than 100 models, very few got externally validated. And again, which ones got validated just with areas on the DRC curve would put this number even smaller. So it is currently an issue in our area. Beyond that, so you can do everything right, but the clinician may decide not to use it, doesn't understand what the model is, doesn't like it, and so on. So that's another thing. Also, is there a control group? Because if you put your system out there and people start relying on it, you would never have the what if I hadn't put it there, right? Maybe the practice was changing in some way or another. So there is no reason not to have a control group for this particular model. It should be considered a device. And then finally, just saying polygenic risk scores is another thing that is coming up, and we have to take all this things into account. We're very interested because we're bringing the Center for Admixed Science and Technology here to UCSD. And what does it mean? We have to check when to use genetic findings in the clinic, and many things are not helpful. They're recalled, like findings that are recalled later. They don't apply to a lot of the patients. And we want to account for the admixed population, because many of the polygenic risk scores will not apply to this population. Again, because a trait is a function of genetic determinants, both variant effects and ancestry, and social determinants, environments, and other things. We think that's the formulation we want to use and the data we want to use without moving data around. But again, if you think the number of variables here, you can imagine the number of observations that we need to have for this model. And our vision is that once we have those kinds of data, we don't need the auto reported race and ethnicity, because those constructs are trying to encapsulate too much in them, ancestry plus the diet plus culture plus other things. But if we can decompose and use them, then it should be available to everyone. So that's what we want to do, and we'll need new methods for that. So one of the things, this is a product of many different areas that we were working on. And I'll leave you with this image. So that's the only image you get in my presentation here. This is a search, stock images for kidney disease. And that's what you get. So thank you so much. I don't know if we have time for a question or two.
Yeah, we have time for questions. She and I were talking just before. So we'll encourage the students to ask the question aspects. This is probably the safest environment we'll ever be, supposed to a giant conference. So not that anybody's going to respond. So questions for
Or opinions. You can have opinions too.
This is the post-COVID, nobody has questions, basically.
Yeah.
Do you think there will be any additional data privacy issue when it comes to imaging? I mean, you talked about being able to reconstruct a face from the genome, but sometimes if we do brain imaging, the face is the data, right? You have that information there.
Well, there have been several papers, I believe, to when you do images of the head that you can reconstruct, and then people try to blur them a little bit and so on. But then if you keep the bone structure there, then people can still do it and so on. So yeah, there are several privacy issues involved. I'm very sure.
I'm not a student though.
Yeah,
Oh.
you were.
It's okay.
So thank you very much. And you encouraged us to get involved if we were interested in bringing some imaging data to all of us. I guess I wanted to ask you a little more, in what form could that involvement take? We have various, imaging data repositories. We gather lots of data, but what would be the best way to interface with all of us?
I think it would be, now that we're bringing the recruitment here, if we could add in the same spirit as the biobank, right? Some image collection in parallel with the biosample and with the electronic health record and the survey. I think we would be the ones to have, again, think about it, you get free sequencing and these other things, and then you add the image. It would be a very valuable resource.
Are there sort of regular fora that go on within all of us where imagers might be able to come in and talk about what's possible, or do we contact you, or what's the best way to get involved?
Yeah, right now I think we have to convince them that we should do it and how we should do it, and probably would be a combination, right, of institutional investment plus all of us approving and coming up with a protocol. But as far as I can tell, there is no protocol to date. And that's one thing that people have noted, because since the UK Biobank, which is 500K people, has the imaging, why can't we go in the same direction? So it would be, again, let's convince our leadership that that's something we could do here.
Thank you.
Thank you. This is really inspiring. I'm so glad you talk about, data sharing strategy and so on, and talk about several things. For example, do federated learning and all those things. Technically speaking, of course, federated learning is definitely the, fancy and good to go. I have no issue. I really like that. But, when we really talk about things is when we contribute, pull all the data together, the current machine learning community, one of the major challenges is whenever we write down a loss, we say, okay, we try to learn from this. Write a formula that is mathematical expectation. And when we do the training, we say, okay, we replace this mathematical expectation by sample mean. But the problem is when we do that, we did not take care of the prevalence, the data prevalence for disease prevalence, and so on. The statistics in the data has been totally removed. So when we do the federated learning, that is essentially do the sample mean. That is no problem. But down the road, in future, if we have good machine learning strategy, really help us to take care of the data distribution, and then, to allow everybody to just contribute the data may not be the way to go. So I want to hear whether you have some even broader vision and thoughts, and how do we eventually address this? I think this fundamental issue.
So let me see if I can simplify the question a little bit. What you're saying is because we don't sample the population, we will have a convenience sample in which we get this hospital plus this one, and you're totally right about that. So there are questions that we cannot answer, right? There are questions that we should not try to answer. It's like prevalence, because we're not doing a random sample of the population. However, some associations you can, right? Given that you have hypertension and you're hospitalized, the rate of death taking this medicine versus taking that medicine, if you see a difference, then you're comparing the rates. You're not comparing kind of the absolute number, right? So with any collection that is not what we call not representative of the population, meaning it's not preserving the distribution that you would find in the population, I think that the number of questions you can ask is more limited for sure. Which doesn't mean you shouldn't do it, because otherwise some minority groups, and like I mentioned, the mixed individuals would never be included, right? So I think there is a movement or a realization that personalized medicine, precision medicine is for everyone. It's not just the majority and the large statistic. So I hope I answered your question.
Yeah, thank you.
Thanks, Lucila. Hi. So you mentioned ROC curves, and I mean, these came out of World War II, right? Radio operators and all this looking for subjective places where performance curves could work. And I think it got adopted by the radiology community in large part, and maybe people in this audience are into it, because, you know, how can reconstruction help a radiologist subjectively pick things out of an image? But as we move towards more and more quantification and bring in things like multi-omics, maybe subjective measures are not the way to go. So we need more true quantitative measures of, you know, here's the tumor, it's in this location, it's precisely this measurement, it's this big, it has these properties. So, I mean, from your world, like, what do you think about that? Or are things getting more quantitative in general, or is it always the human observer at the end that the ROC curves try to mimic in some way, like coming up with ideal likelihood observers, but maybe there needs to be something more completely quantitative and not so human observer oriented?
Yeah, and I didn't input the details, but many of these are quantitative prediction models that estimate a certain probability, and then you build the ROC curves out of them. And it's this plague, I would say. Whenever a big paper comes and uses particular methods, the other people start using it because, yeah, after all, it got accepted in Nature or Science and so on. And sometimes it's not the best, right? But AUC in this case is very, if you take any machine learning person, that's the one thing they know it's there, right? But the calibration part or other things that errors, again, I think errors would be much more indicative of the quality of the model. But it's sometimes, yeah, somehow it got trendy or acceptable for reviewers to use areas in the ROC curve, and it went that way. But I mean, I don't want to diminish their value either, because the radiology community invested so much in them, and it has very good uses. It's just not the absolute and the ultimate evaluation method.